All right, so welcome back, and today we're going to be looking at the structure of nucleic acids, so DNA and RNA, because they'll both be very important for our topics here in this unit of DNA replication and protein synthesis. Of course, DNA is very important in DNA replication, and RNA takes kind of the center stage for protein synthesis. So, I know way back the beginning of the year when we had biochemistry, the chemicals of life, we talked briefly about the structure and function of nucleic acids, but now it's going to really come back to be in the center stage. So we want to go over that a little bit more explicitly. So remember, the function of nucleic acids is that they serve as the genetic material, the material of inheritance. So they store the genetic information in the case of DNA, store those alleles. Um, and the molecules of DNA are made up of many genes. So that is those sequences of bases that help to produce a given protein. So DNA stores genetic information in genes, and they provide, like I said, those blueprints, those instructions to make proteins. So we have this kind of flow from DNA to RNA to proteins. This is called the central dogma of biology. And so DNA is kind of the ultimate source of that genetic information, and RNA is kind of the intermediary between the blueprint and the production of proteins themselves. So here's kind of a pictorial representation. We've got the double helix of DNA that contains all of the genes that store the instructions of how to make different proteins, and they get turned into, and this is a space-filling model of a protein. Nucleic acids also transfer information, and that's kind of the role, both of RNA transferring information in DNA to proteins, and of DNA in new daughter cells, transferred the genetic information from parents to offspring in the case of multicellular organisms. So each of these two daughter cells will have identical copies of DNA just like the parent cell, and offspring will have a combination of parental DNA in the case of sexual reproduction. So, nucleic acids, the big examples, or the only examples we're going to talk about, is RNA, which is short for ribonucleic acid. And there's a picture of RNA, noticing it's single-stranded. And DNA, deoxyribonucleic acid, and there is a picture. So you can kind of see the macro structure of both from those images. And DNA, of course, from those images are double helix. So really, even without looking any closer, we can see one huge difference between the two. But in either case, and if we think back to biochemistry, they're made up of repeating units or monomers called nucleotides. So each of these are made up of, as you can see, like each of these rungs. You can see on either side, these are the nucleotides. And we'll look at nucleotide structure in just a little bit. So a nucleotide is the monomer, the repeating part that makes up all nucleic acids, and it's composed of three parts: a nitrogenous base, or a nitrogen-containing base. It's that part right there. These can come in 5 different varieties. So the A, T, C, G, or U, the adenine, thymine, cytosine, guanine, or uracil. Uh, they also need a pentose sugar. That just means a 5-carbon sugar. So, like 1, 2, 3, 4, 5. Pentose sugar. So all nucleic acids, nucleotides have a pentose sugar, whether it's deoxyribose or ribose. So in RNA, Ribose is the specific pentose sugar that is present, whereas in DNA, deoxyribose sugar is. That's the part that you need to know. You don't need to like identify from a picture like this whether it is ribose or deoxyribose. But here you can see, if you are interested, you can maybe look at these and notice the striking similarities and subtle differences between the two. And then lastly, both contain a phosphate group. So PO4, that's right over here that you can see on this model. So right there, you can see a nucleotide, and you can see the polymerization of that nucleotide into the start of a nucleic acid, whether it be DNA or an RNA. So here is a picture of, again, the structures of DNA and RNA side by side, a kind of an artistic representation, and you can compare kind of based on what we talked about, the similarities and differences. Each of these ladder rungs represent the bases, and then the backbone or the side parts represent the phosphate group, pentose sugar kind of combined together. So we can group the nucleotides into kind of two types that is concerned with what nitrogenous bases that nucleotide has. So if there is a nucleotide that has either an adenine or a guanine nitrogenous base, we call those the purine bases. Or if there is a nucleotide that is that contains the cytosine or the thymine nitrogenous bases, we call that a pyrimidine. And you can see here the purines and the pyrimidines. And the purines are all double-ringed bases. So here's adenine. It's got two rings as part of the adenine base. Same thing with guanine. And the pyrimidines all have a single ring. So A and G are the purines, and C, T, and U are the pyrimidines. There's U. I thought it was going to be on there somewhere. So here is another picture of the same thing, noticing that uracil, U, is only present in RNA, and thymine T is only present in DNA. Actually, these two replace each other, so U takes the place of T in RNA, and takes the place of U in DNA in either way. And you can kind of see how that's incredible of how strikingly similar they are between these two. All right. Here we compare the sugars that are in DNA and RNA, so deoxyribose versus ribose, and you can see the subtle differences between the two. Note that that's not going to be, a question that I would ask to circle deoxyribose, but I thought it would be interesting to to show that, the whole difference comes down to very, very few different atoms that are different. And then finally, we get the nucleotide polymers, so making the whole nucleic acid out of the nucleotides. So in both cases, adjacent nucleotides are joined by covalent bonds between the OH group on the 3 carbon of one nucleotide and the phosphate on the 5 carbon of the next one. And I'll show you that in a picture coming up. Those linkages, which happen via, remember, dehydration, removing water synthesis, create that backbone of the sugar phosphate with those nitrogenous bases coming out in between like ladder rungs. And the sequence of bases along a DNA or RNA polymer is unique for each gene. So each gene is a different combination of maybe tens or hundreds or thousands of different nucleotide combinations. So here we have a picture of our DNA, and here let me just go back. So what we were saying previously on the previous slide, is if we look at our nucleotide, we remember that up here this is the oxygen, and we count 1, 2, 3, 4, 5. So this is our 3 prime, or wait, just a second. That's not correct. So we have the 1, 2, 3, 4, 5. There we go. So 5 prime and 3 prime. So if we go back, like it says, we polymerize our nucleic acid um, by joining covalent bonds between the OH group on the 3' end to one of the nucleotides and the phosphate on the 5' end. So right here is where that polymerization occurs. We add this bond to join these. So if this was only, let's say this big one nucleotide, we'd add the next and it would float right in here and this bond would form and then we'd add this on. And then this one would float in and we'd add this on. So the sugar PO4 bond, so that one that I was just highlighting right here, is called a phosphodiester bond. That's the technical name for this right here, a phosphodiester bond. We've got 1 phosphodiester bond, 2 phosphodiester bonds, 3, 4, and that's where the new base is added to the sugar of the previous base. This is, like I said, this is the bond that's being formed in that polymerization reaction, so that our polymer, our nucleic acid, is only growing kind of in this end, this way. It only goes in one direction. Okay, we add it going from, if you look, we add it 3 prime, 5 prime, going in that direction. So kind of again, to summarize, RNA molecules usually only exist as single polypeptide chains. I say usually because in some viruses, can actually have a genetic information of double-stranded RNA, which is really interesting. DNA molecules have 2 polynucleotides spiraling around an axis, forming that double helix shape, right? And in the DNA double helix, the 2 backbones run in opposite directions. So remember, they're anti-parallel. If this is DNA, and this is one strand of DNA, the other strand over here would be going the opposite direction, where everything would be pointing the opposite way, and so it would be going, from 3 prime this way to 5 prime that way, and that's called anti-parallel. Or you could just draw, the symbol anti-parallel like that. Lastly, if we look at the pairing of the nucleotides, the bases that are able to bond with each other are always the same. So hydrogen bonds form between complementary base pairs, and that's what stabilizes the DNA molecule. So adenine and thymine are always paired together, and cytosine and guanine are always paired together with hydrogen bonds. And notice how 2 hydrogen bonds can form between adenine and thymine, whereas cytosine and guanine, 3 hydrogen bonds can form between them. So this is how the 2 strands, anti-parallel, end up matching up and staying together and not just floating apart, as those hydrogen bonds that form. And it's always these same pairs that will bond together. C will never pair with A, or G will never pair with A or anything. It's always A, T, C, and G. A, T, C, and G, just like that. So again, here is our last image of a DNA molecule, showing it in its double helix shape, and showing you again in the dotted lines representing the hydrogen bonds. Okay, so hopefully that was a good refresher kind of about the structure of DNA and RNA. We'll be dealing with excuse me, them a lot more in this unit, but that was just, to get the very pertinent information to use as we get started.